Hello everyone. I'm Ashlishya. In the summer, I have been working with the Bridges doing research in the biochemistry department. The title of my poster is Utilizing Advanced Tool to Analyze Breast Cancer Mitochondrial DNA Mutations. Breast cancer is the second most common cancer in women, especially in the United States. It is a condition in which the breast tissues proliferate uncontrollably, making a lump-like structure, which is the tumor. According to its origin, it can be classified into two different things. If it originates from the duct, it is known as invasive ductal carcinoma, whereas if it originates from the milk gland, which is also known as lobules, it is known as invasive lobular carcinoma. Studying human mitochondrial DNA replication is really important to look at what disease it can link to and trying to find a solution for it. So in this poster, we are investigating into two different type of thing. The first thing we're investigating is to try to find a tumor-specific breast cancer ND3 mitochondrial DNA mutations of all the breast cancer data we have. In addition to that, we are also looking into making a row zero cell using our mouse cell line, which is C2C12, to study the mitochondrial DNA replication. So row zero cells are cells which do not have their mitochondrial DNA when they are exposed in a mutagen like ethidium bromide over a longer period of time. As I've mentioned before, we are looking into two different things, so we have two research questions followed by one hypothesis of each. The first research question is, are there ND3 tumor-specific mitochondrial DNA mutations in breast cancer? So we assume that yes, we will try to find some tumor-specific mitochondrial DNA mutations which can alter the cancer cell metabolism. The second research question is, can C2C12 cell be depleted of mitochondrial DNA? And we assume that yes, it will deplete their mitochondrial DNA. So this picture over here is the human mitochondrial DNA genome, and I have only worked with this specific part, which is ND3 for the breast cancer data analysis. For the method, the first part was the lab work of making the row zero cells. Proliferating C2C12 cells were seeded in a tissue culture dish. Ethidium bromide was added after a few days. They were grown for 6 weeks. In that long period of time, they were splitted when they were 80% confluent, and media was changed after every 3 days. Then they were transferred to a 96 well plate where they were grown for 2 weeks, and we found two clones. According to their well number, we named them G6 and G7. Those cells were harvested, DNA was extracted and quantified, PCR was run, and those PCR products were loaded in a 1% agarose gel to find, and the gel was imaged under G-box to find out and determine the result. The next part of the materials and methods was the analysis part of the breast cancer. So this website, Guhilet, is founded by one of our co-author, which is Dr. Ravi. So in this website, first we have to search the variant position in this box, which gonna lead to this web page where we're gonna look into three different type of thing. We're gonna look the effect, whether it is synonymous or non-synonymous. We're gonna look whether it's present in haplogroup or not, and third, we're gonna look its variant. So then we're gonna go to this link, which is MitoMap, which leads to the next page where we're gonna search the position with the variant and try to see information. So we're gonna see at the apogee part because it is a tool which tells whether it's pathogenic or not. And then we're gonna look into the references over here, which are listed, and try to find a paper which tells like detailed information about that specific mutations. This leads us to the result. In the result, the first table is for the breast cancer analysis. So variant is what we got from these website. Cohort, it tells about, so our data has three different types. It have from Norway, Guhilet, and Sweden. So I have used only the Sweden data. So it's written 0y0 because it's not present in Norway, not in Guhilet, but only present in Sweden. And in the cohort, it's all of them SWD, which stands for Sweden, and have this NT. So if one NT means that one mutation was present in this Sweden patient, two NT means the same mutation was present in two people in the Sweden data. So these are the notes which we took by looking at the references. Unfortunately, we couldn't find any tumor-specific mutations because I only worked with this little ND3 part of the cohort, and there is a whole lot to work with, and we had a limited period of time to work with. The next part of the result is the gel image. So our gel have 7 lanes, which is gene ruler followed by G6, G7, a negative control, negative control, and gene ruler. So gene ruler is given to the first and last lane because it works as a ladder to show what each base pair can look like. So lane 2 and 3 was G6 and G7 clone. Unfortunately, we can see those black lines over there, which tells us that the mitochondrial DNA was not depleted. So the reason behind this could be that we need to use a different concentration of ethidium bromide to work with, or we should grow them for a longer period of time. For my future study, I would like to study a different cell line, which is 143TKB. It is a human cell line, and there have been many papers who worked with it and were successful of making row zero cells. I really appreciate the opportunity I got from SI Bridges, funded by NIH, and all my co-authors who worked really hard to do this research with me. Here are my references. Thank you.